Hello dear students, welcome back again to Chinmoy's Biology channel and welcome to yet another new episode of tissues. Let us start with plant tissues first and then we will move on to animal tissues. So let's get started without further delay. So in the last chapter, we can recall that living organisms are made up of cell, right? So we have studied in details about cells in the previous lesson. So I think you remember that, and if you don't know the previous lesson which I have taught, so I'll be putting the link below. Now, in unicellular organisms, a single cell performs all basic functions. So, in a single cell, only all basic functions are there in a unicellular organism. So, for example, in amoeba, which is a single cell which carries the movement, intake of food, gaseous exchange and excretion. But the multicellular organisms, there are millions of cells. Okay, so not only us, there are other multicellular organisms as well. So we are having millions of cells. So most of these cells are specialized to carry out specific functions, right? So each specialized function is taken up by the different group of cells. So this different group of cells are formed to perform different functions. So since these cells carry out only a particular functions that they can do efficiently, in human beings, like muscle cells contract and relax to cause movement, nerve cells carry messages, blood flows to transport oxygen, food and hormones and other waste materials and so on. So like that, only different cells here we can see that as in human beings, different cells are specified for different functions, right? So like that, only in plants, vascular tissues conduct food and water from one part of the plant to other parts. So multicellular organisms show division of labor. So it means that each bunch of cells will be performing a specific function or a specialized function. So cells specializing in one function are often grouped together in a body. So this means that a particular function is carried out by a cluster of cells at a definite place in the body. Now this cluster of cells is called a tissue, right? Is arranged and designed so as to give the highest possible efficiency of function. So blood, phloem and muscle are all examples of tissues, right? So the cluster of cells, this cluster of cells called a tissue is arranged and designed so that it is giving the highest possible efficiency of function, right? So like blood, phloem, muscle, they are all tissues and they are performing a specific function with its highest efficiency. So is it clear? Now a group of cells that are similar in structure and work together, they are called form a tissue. So what is a tissue? A group of cells that are similar in structure or they are working together to achieve a particular function is a tissue. So this is the definition of tissue, right? So are plants and animals made of same types of tissue? So this is a very important question. Are plants and animals made up of same types of tissue? So let us now discuss about the tissues in plants and animals. So do plants and animals have same structure? Do both of them perform similar functions? No, it's not like that. So both plants and animals are having different types of tissues, but they are different. So what are they? So there are noticeable differences between the two. That is, plants are stationary or they are fixed. So they can't move from one place to another. So since they have to be upright and they have a large quantity of supportive tissue. So what is to support them? Right, so they are standing in one place and they are upright always. So the supportive tissue generally has dead cells. So that will be a different kind of cell. Animals, what is the criteria of animals? They on the other hand move around in search of food, mates, shelter. They consume more energy compared to plants and most of the tissues they contain are living. So in animals, mostly the body is composed of living cells, whereas in plants it is composed of dead cells, right? Now another difference between animals and plants is the pattern of growth. The growth of plants, they are limited to certain regions only, while This is not in animals. There are some tissues in plants that divide throughout their life, right? There are some tissues in plants that keep on dividing throughout their life. These tissues are localized in central regions, right? Based on the dividing capacity of the tissues, various plant tissues can be classified as growing, meristematic tissue or permanent tissue. So the tissues which are localized in the central part, they have the dividing capacity and they can divide throughout the life cycle of the plant. That is, they are as growing as a meristematic tissue or a permanent tissue. Now, cell growth in animals. So this is a difference. Cell growth in animals is more uniform. So there is no demarcation or dividing or non-dividing regions in animals. Okay, so this part, the central part only will grow, the other parts will be remaining the same throughout the life. No, it's not like that. There is no division or demarcation of division in an animal body, right? It is throughout, it is dividing in the same manner. The structural organization of organs and the organ systems is far more specialized and localized in complex animals and even in very complex plants. So the structural organization in a complex animal, it is also localized and specialized as well as in complex plants. So the fundamental difference is what? It reflects the difference of modes of life pursued by the animals and plant organism, right? Particularly in the different feeding methods. So like that only the tissues will be varying. Now also they are differently adapted for a sedentary existence on hands plants and active locomotion on the animals. We have already told earlier, because the plants are upright, they have a sedentary life, they are not moving from one place to another, they are having mostly dead cells, whereas animals who are in active locomotion, they will have or contribute to the living tissues, okay, contributing to different design of organ system. Okay, so with this reference of complex animals and plants body, we will talk now about the concept of tissues in detail. So all tissues, first we will start with plants, then we will move on to animals, right? Now, first two questions are there. This I have already discussed earlier. You should write the answer in your answer book. What is a tissue? What is the utility of tissues in multicellular organisms? I have already told that they are performing a specialized function with their full capacity, or with they are actually these group of cells or tissues, they are specialized for a perfect particular function, right? So what is the utility of tissues in a multicellular organism? That these two answers you can write it on your own. I have already discussed. Now let us start with plant tissues. Now first, in plant tissue we will take the meristematic tissue. Now here we start with an activity. Now here in activity, let us see, we are taking 2 glass jars and we are filling them with water, right? Now take 2 onion bulbs and place one on each jar as shown in this figure. Now just you are dipping them, observe the growth of roots and bulbs in a few days, right? Now 2 glass jars, you have filled them with water. Now you are placing the onions like this and you are seeing the growth of this roots and you are measuring them, okay, on day 1, day 2 and day 3, right? On day 4, what you are doing, you are cutting the tips of onion bulb in the jar by about 1 centimeter. So here you are cutting the tips by about 1 centimeter in day 4. So after this, observe the growth of roots in both the jars and measure their lengths each day for 5 more days. So this is the table. Here you are supposed to write as per your observation what is happening in jar 1. Jar 1 was kept intact. You didn't cut 1 centimeter their root, but jar 2 you have cut the root for 1 centimeter, right? So then how the roots are growing, what is the growth rate from day 1 to day 5 after that cut is being done, you are observing, right? So let us see what will happen. Now from the above observations, answer the following questions. Which of the 2 onion has longer roots? Why? Do the roots continue growing even after they are removed from the tips? Why would the tips stop growing in jar 2 after we cut them? Just go through the observation. It is very easy observation. and you'll just go through it, right? Now, the growth of plants that occurs only in certain regions. These specific regions are there where the growth happens in plants, not in all the parts. Now, this is because dividing tissue, which is known as the meristematic tissue, is located only in these points itself, right? So, where depending on the region where they are present, meristematic tissue, it is on that basis, it is classified as a apical meristem, lateral meristem, or intercalary meristem, right? So, that we will read now. Now, new cells produced by the meristem are initially like those of the meristem itself, right? So, as the meristem was initially like those only, the new cells will be produced, but as they grow and mature, their characteristics slowly change and they become differentiated as components of other tissues, right? Now, here you can see the meristematic tissue, when it is present in this apical region, it is called apical meristem. When it is present here, it is called the intercalary meristem, and when it is present at the sides, it is called the lateral meristem. Is it clear? So, depending on the location where it is located, it is defined like that only: apical meristem, intercalary meristem, or lateral meristem. Now, apical meristem is always present at the growing tips of stems, roots, and it increases. The length of the stem and the root. The girth of the stem or the root increases due to the presence of the lateral meristem or the cambium. So, as the lateral meristem is present, it will increase its girth. Okay. The intercalary meristem seen in some plants is located inside near their node. Okay, near their node means here is a node present. So, this near the node, this intercalary meristem will be present, right? So, these all are helping the specific growth in that region, right? The cells of meristematic tissue are very active. They have dense cytoplasm, thin cellulose cells, and prominent nuclei. So, what is the characteristic of meristematic tissue? They have dense cytoplasm, they have thin cellulose walls, and they have prominent nuclei. They lack the vacuoles, they do not have vacuoles, and can we think that when they would lack vacuoles, you might want to refer to the functions of vacuoles on the chapter of cells. I have already discussed about the functions of vacuoles in the previous lesson. Please do refer to it, as I'll be posting the link in the description box as well as in the I link above, right? So please go through it. Now we will go to the permanent tissue. Now, starting with this permanent tissue, what happens to the cells formed by meristematic tissue? Now, cells have been formed by meristematic tissue. What they are doing? They take up certain specific role and lose the ability to divide. So, they are losing the ability to divide, and as a result, they form a permanent tissue, right? So, this process of taking up a permanent shape, size, and a function is called differentiation, right? So, this process of taking up of a permanent shape, so they are taking a permanent shape, they are taking a permanent size and a function, which is called differentiation. So, differentiation leads to the development of various types of permanent tissues, okay? So, as it is taking a shape, size, and structure and having a permanent function, so this process is called differentiation, right? Now, let us choose an activity and just take a plant stem with the help of your teacher and cut it into thin slices or sections with the help of a blade, or we can say a knife. So, it should be very thin. Now, stain the slices with saffron in solution. It will be red colored saffron in, and neatly cut the section on the side and on a slide and put a drop of glycerin on it. Now you can mount a cover slip. Just learn these things from your teacher. You can mount a cover slip. You can observe under a microscope and observe the various types of cells and their arrangement. Right now, you are comparing in figure 6.3 that are the cells similar in structure? How many cells are being seen? Now you can see in this 6.3. See, are the cells all similar in structure? No. So how they look in this structure, are they all similar? No. How they look, what are the different layers? So can you think of reasons why there would be so many types of cells? So there are so many variety of cells in that stem cutting you have placed on the under the microscope. So we can also try out cutting sections of roots. We can even try cutting sections of root and stem in different plants. Now first we will start with the simple permanent tissue. Now under the permanent tissue, there are simple permanent tissue and complex permanent tissue. Now we will start with simple permanent tissue. Now a few layers of cell beneath the epidermis. Now this is the epidermis or the outer layer. Below this epidermis, a simple permanent tissue we can see just beneath the epidermis, few layers below the epidermis, we can see the parenchyma is the most simple permanent tissue, common simple permanent tissue, and it relatively consists of the unspecialized cells with thin cell walls. So they are unspecialized cell, they are having thin cell walls, they are all living cells. So what are they? They are usually loosely arranged. You can see they are loosely arranged. These cells, parenchyma cells, they are all loosely arranged, and they are having large spaces in between the cells, that is, they are having intercellular spaces. So let us zoom into the diagram. So these are all parenchyma cells. They are loosely packed. They are having fine outer cell wall. We can say that their outer specialized cell walls are very thin. They are not specialized at all. They are unspecialized. They are having intercellular spaces in between them. So they are not tightly packed, and they are having intercellular spaces in between them. Next comes this tissue generally stores food. So parenchyma function is storing food. Now in some situations it contains chlorophyll. When the parenchyma is having chlorophyll, it performs photosynthesis function. It is then called chlorenchyma. Storing chlorophyll, it is performing photosynthesis, it is called chlorenchyma. In aquatic plants, the large air cavities are present in parenchyma to help them float. So when they are having air cavities in them which is helping them to float, such a parenchyma type is called the aerenchyma. So which is helping them to float is called the aerenchyma, right? Now the flexibility in plants is due to another permanent tissue that is called the collenchyma. So here you can see this one is the parenchyma. Parenchyma, if it is having chlorophyll, it is called chlorenchyma, and it is having air to help it float, it is called aerenchyma. It is clear, it is very easy. Now there is another type of simple permanent tissue, that is collenchyma. Now we are going to discuss about these cells, collenchyma. Now it allows the bending of various parts of the plant like the tendrils, stems of climbers without breaking them. So that flexibility which helps the bending of the tendrils, the parts of the stems or climbers, that is due to the collenchyma. It provides the mechanical support. We can find this tissue in leaf stalks below the epidermis. So just below the epidermis and leaf stalks, it is in the section of a stem also, just below the epidermis is the collenchyma cells. So the cells of this tissue are living, elongated and irregularly thickened at corners. This is very little intercellular space is present. So now let us see first parenchyma we have seen, then collenchyma. Collenchyma is that wall thickenings, you can see it is irregularly thickened. Some places it is more thick, some places it is less thick. So irregularly thickened, they are having a nucleus, they are having vacuole, and they are having a cell wall. So parenchyma, you know what is collenchyma, sorry, it is different from parenchyma. So I have told you the differences. Now we will move on to yet another permanent tissue, that is the scherenchyma. So scherenchyma, it is the tissue which makes the plant hard and stiff. So this is scherenchyma, which is making the tissue hard and still. We have seen the husk of a coconut. It is made up of scherenchymatous tissue. The cells of this tissue are dead and they are long, narrow, as walls are thickened due to lignin. So these thickened walls are due to lignin. So thick lignified walls. Scherenchyma characteristic is thick lignified walls. So often these walls are so thick that they are having no internal space inside the cells, and this tissue present in stems around the vascular bundles, in the veins of leaves, and in hard covering of the seeds and nuts. So where it is present? It is mainly present in the hard coverings of seeds, nuts, and coconut shell. So these are lignified, hard, thick walls. Okay, and they are mainly the dead cells. The cells of this tissue are nearly dead. Okay, so we will now move on to one activity. Now let us start with an activity. Now take a freshly plucked Rio leaf. Now that plant, I'll show you what is a Rio plant in the next slide. Stretch and break it, applying pressure. While breaking it, keep it stretched while gently so that some peel or skin projects out from the cut. Okay, so you press and break it in such a way one part of the skin moves out. So remove this peel and put it in a petri dish filled with water. Add a few drops of saffron in, wait for the couple of minutes and then transfer it to a slide. Gently place a cover slip over it and observe it under a microscope. So what you are observing? When you observe, the outermost layer of cells is called the epidermis. The epidermis is usually made up of single layer of cells. Now in some plants living in dry habitats, the epidermis may be thicker. Since protection against the water loss is critical, so epidermis has become thicker so that transpiration happens less, and the outer layer is a protective layer. The entire surface of the plant has an outer covering epidermis. It protects all the parts of the plant. Epidermal cells on the aerial parts of the plant often secrete a waxy water resistant layer on their outer surface, right? So this aids in protection against the loss of water, mechanical injury, and invasion of parasitic fungi. So what is happening? This resistant layer, this epidermal thick layer, it is the outer surface. It is protecting against the water loss, against the mechanical injury and invasion by the parasitic fungi. Since it has a protective role to play, cells of the epidermal tissue, they form a continuous layer without any intercellular spaces. So they do not have any intercellular spaces. Most epidermal cells are relatively flat in nature, and often their outer and side walls are thicker than the inner walls. Their outer and the side walls are thicker than the inner walls. We can observe small pores here, and there is epidermis of the leaf. These small pores are called stomata. You can see here. You can see this. These are epidermal cells. These are the outer layering cells on the outer layer of the leaf. And these epidermal cells, they are having stoma or stomata. And these stomata are often guarded by two cells called the guard cells. See this is the stomata or the opening of the stoma, and these are the guard cells on the two sides of the stomata. Okay. Now stomata are often enclosed by two kidney shaped cells called the guard cells, and they are necessary for exchanging gases with the atmosphere. Transpiration, loss of water in the form of water vapor also takes place through the stomata, right? So transpiration, loss of water in the form of vapor, also it's taking place through the stomata. Now we will recall which gas is required for photosynthesis. Find out the role of transpiration in plants. So which gas is required for photosynthesis? Carbon dioxide, we all know. And find out the role of transpiration in plants. Now the epidermal cells of the roots, what they are doing, whose function is the water absorption, they commonly bear long hair-like parts. Now before moving to this, I want you to show you the Rio plant. Let us see that. Now here you can see this is the Rio plant. This is having thick leaves one of these leaves only you are taking and you are trying to press and break it so that the outer layer or the epidermal layer comes out, which you are soaking in a petri dish in a saffron solution, and you are placing it under the microscope to see the cells present in the epidermal cells of this Rio plant. This is basically an indoor plant and it is mainly staying in dry habitat, and it is a plant which is shade loving. It is okay to keep it indoors, which will add a beauty to your rooms, as well as no need to take it out in sun and all. So it can stay well. So once in a while you can take it out and then again you can place it indoors. So now we will move on to the epidermal cells of the roots which we were discussing. Now, the epidermal cells of roots whose function is water absorption, commonly they bear long hair-like parts that greatly increase the total absorption surface area. Like in some plants, like desert plants, epidermis has a thick waxy coating of cutin. So it is having a waxy coating of cutin, chemical substance with waterproof quality on outer surface. Can we think of a reason for this? Yes, in the outer surface of the branch of a tree, the different from that of the outer layer of a young stem. So why is the outer surface of a branch of a tree different from the branch of a young stem? So as plants grow older, what is happening? The outer protective tissue, it undergoes certain changes, because when the plant is young, it is having soft stem, but as it grows older, it is having hard stem, right? It undergoes certain changes. A strip of secondary meristem located in the cortex forms layers which constitute the cork. So cork is also made up of dead cells. So it is strong, it is hard, it is having a protective power. Okay, so the cork are dead and compactly arranged without any intercellular spaces, and they also have a substance called suberin in their walls that make them impervious to gases and water. So what is happening? They are not, they are hard, they are compactly arranged, and they are impervious to gases as well as water. So water will not enter, gases will also not enter through this protective layer, through this hard stem. So this is the ruptured epidermis. If the epidermis is ruptured also, here are a bunch of cork cells inside that. So below just the epidermal layers, these cork cells or the cambiums are having substance called suberin, which is protecting the stems from the gases as well as the water. So this is all about the simple permanent tissue. Okay. Now the complex permanent tissue again is a bit different from simple permanent tissue, and this we will be studying in the next class, because or else we'll mix it up. We have today studied about the permanent tissue, that is simple permanent tissue. In that, parenchyma, collenchyma, scherenchyma we have studied, and we have studied about aerenchyma also. Okay, so we have studied the structure of the cells, we have studied how they look like, and if you are placing under a microscope, how this differences between the cell of collenchyma, parenchyma, scherenchyma, and transverse and longitudinal section will look like. And the permanent tissue we will be reading in the next class. Complex permanent tissue. Okay, so stay tuned with all the episodes which I am posting. Don't skip the lessons in between, because throughout the episode I'll be teaching you many important things in details. So don't skip. Go throughout my lesson, and please do subscribe to my channel so that whenever I am putting my updates, you will be getting it first. And please don't forget to share it with your friends. So stay tuned, stay updated with all the lessons which I am posting. Thank you, and we'll be meeting you very soon with the complex permanent tissue. Thank you.